Okay, so the last couple of presentations we've been talking about photosynthesis, how it's broken down into the light reactions, and I like to try to emphasize light-dependent reactions, and the dark reactions, or light-independent. Today, we're going to look at some alternate pathways because under different conditions, photosynthesis is going to occur at different methods. Now, one of the big problems with evolving life on Earth is dehydration. Before plants could evolve and evolve onto land into the various forms that we see, they had to overcome the problem of dehydration. Okay? Now, in dehydration, there's remember those little tiny holes at the bottom of the leaf, the stomata? Well, if they're open to allow carbon dioxide to come in, they're also going to allow water to leave. Water vapor to leave. So leaving those stomata open all the time, is going to allow the plants to dehydrate much quicker. Now, other plants have evolved different ways to combat this. Think about the pine trees and their needles. Thick waxy coat on the outside, very small surface area. So less chance to dehydrate in the cold, dry winter breezes. But just getting onto land, going out of the water, when you're trying to move farther away from the water, dehydration is a big problem. Now, the solutions that they've come up with, now again, evolution is not driven, just whatever works to better that organism's chances to survive is going to be passed on here. Some of the solutions, may seem to conflict with photosynthesis. As we look through these three methods here we're going to be looking at here in a little bit. And again, like I've said earlier here, the stomata, while they're great to let CO2 in and oxygen to leave, they also cause a lot of water to evaporate. So on hot, dry days, plants will close the stomata. That's going to conserve water. But when they close the stomata, that's going to interfere with photosynthesis because close the stomata, and there's not going to be as much CO2 to come in. And also, oxygen is not going to leave. Now, the problem with that, as we'll look at the next slide, is what happens when oxygen levels get too high because the plant closes the stomata. It's a major problem here for the plant. Open the stomata, possibly dehydrate, but you can't undergo photosynthesis. Or close it so you conserve water, but you're going to have oxygen build up on the inside of the cells or inside of the plant's leaves. So this slide here, we just kind of want to point out a few of the big problems, and next couple slides we'll be discussing how evolution has worked to shape the different plants. We're going to begin with a group of plants that basically called C3. C3 plants are all the plants that we've been talking about so far. C3 plants, we've discussed the Calvin cycle, or the light independent reactions. We were talking about C3 plants, where you basically have the carbon dioxide combined with the Rubisco, through Rubisco with RBP, and go through the standard Calvin cycle and produce G3P. Okay, and eventually form glucose. That's all we were talking about. We were talking about the C3 plants. And what happens with these, just like we've discussed previously on the previous slide, really hot, dry days, the stomata are going to close, and any CO2 that's trapped in there is going to be going through the Calvin cycle. But obviously on the inside of the leaf then, the CO2 level is going to drop, but because of photosynthesis, the O2 level is going to rise really, really rapidly. This is what we call photorespiration. Because what happens, normally Rubisco is going to take CO2. But when there's not as much CO2 and a lot more O2, Rubisco can add oxygen to the RuBP. That's what we call photorespiration, and that's going to interfere with the actual respiration because in order to actually make the glucose, you've got to have the CO2 combined with the RuBP. with the enzyme Rubisco. All right? If you're combining it with oxygen, you're going to prompt it to go through respiration. And let's face it, what's going to happen then if it goes through respiration, the plant will produce more CO2 as a result of respiration. So with the stomata closed, you're going to really slow down the amount of photosynthesis. Even though you're going to regenerate some CO2 through respiration, it's not going to be enough to really produce enough food for the plant. But again, C3 plants, that's pretty standard. That's every plant that we've looked at. Our labs here with the spinach leaves, those would be good examples of how the C3 plants operate. Now, while these are the C3s are the ones we always talk about in biology classes, this next one, C4 plants, operate a little differently. Okay, the C4 plants, they're going to first of all fix the CO2 into a 4-carbon compound, hence C4. Instead of taking the carbon dioxide and joining it to the RuBP with the Rubisco, it just automatically fixes it to an alternate 4-carbon compound. So you're going to combine these together, and what's going to happen then, the mesophyll cells are going to take that CO2 and incorporate it into their own organic molecules. So you're going to make a whole new compound here. And the compound, PEP, we just call it PEP, phosphophenol pyruvate, okay, it is added to the CO2, and the enzyme that does that is PEP carboxylase. Now this carbon dioxide is now stored. It's stored in a new form. And this new form, oxaloacetate, gets moved into specialized cells inside the leaf, the bundle sheath cells. These are the cells that surround like the veins. And when they do get down to there, the oxaloacetate is broken back down into carbon dioxide, and boom, runs right back through the Calvin cycle again. Okay, so in essence, everybody, another way to look at what's going on here, rather than these long wordy explanations I put up here, think about this. Okay, and I couldn't think of a better way to put it than very wordy at first. But think about this, that the plants are taking this carbon dioxide, right here, turning it into oxaloacetate, and the oxaloacetate is taken off someplace else, near the veins of the leaf, the bundle sheath cells. And there, the oxaloacetate gives back the CO2, so we have phosphophenol pyruvate to go back again. So all that's really happening, everybody, is that in C4 plants, the carbon dioxide is being pulled away from the photosynthesis centers. And as it's pulled away from there, it frees up the Rubisco to combine with more CO2. Because remember, it's the ratio between CO2 and oxygen that determines whether or not Rubisco picks up the carbon dioxide. If the O2 levels get too high, then it's going to pick up oxygen. And then oxygen has to be converted back into CO2 through respiration before it can actually be used for sugar production. Okay, so by pulling the carbon dioxide away, it gives the plant a chance to increase sugar production. So you're going to see these plants, any kind of C4 plants, are going to be found any place where there's high humidity, or excuse me, high temperatures with lots and lots of sunlight. All right? So plants that you're going to be looking at here, basically like sugarcane, for example, is a good example of a C4 plant. Okay, where it's going to be maximizing sugar production in areas of very intense sunlight and very high temperatures. So really, everybody, at this point, the plant can close off its stomata and still keep pulling that carbon dioxide away, so it prevents the buildup of the oxygen carbon dioxide ratio. So carbon dioxide is always going to be being pulled away and always going to be able to fix to our RuBP and go through the Calvin cycle. All right. Now the third type of plants, or all the pathways for photosynthesis, CAM plants. Okay, fancy name for the acid they're converted into. Okay, CAM plants have the distinction they're operating in such high heat areas here that during the day the stomata close, and at night they open. So let's think about what's happening here. If they close during the day when the temperature is really, really hot, they're not going to lose a lot of moisture. But at the same time, they are not going to bring in any carbon dioxide. They're not going to be able to get rid of any oxygen. And failure to get rid of the oxygen, that's going to lead to photorespiration and a decrease in sugar production. So what happens then, leaving them open at night, if you leave them open at night, you allow the CO2 in. All right? If the CO2 comes in, these plants are going to take that CO2 and fix it into various organic acids inside the mesophyll cells. Don't worry, we're not going to have you guys memorize the names of these different acids. Just that we're going to just understand that we're going to take the CO2 and inside the mesophyll cells turn them into various acids. And that's how we're going to store them. So the CO2 is taken in all during the day, or excuse me, all during the night, fixed into these acids for storage. Then during the day, when the sun comes back up, and the light reactions can supply ATP and NADPH, that carbon dioxide is pulled off those organic acids and runs through the Calvin cycle. I think these are probably a pretty easy way to remember that you're just basically, in CAM plants, you're taking them into acids, and then during the day, acids break down to release the carbon dioxide, and you go through the Calvin cycle. And it's something similar with the C4 plants, where they're just basically shuffled off here to the bundle sheath cells before the CO2 is released again, and we go through the Calvin cycle. But you'll notice that all of these, whether it's the C3, C4, or the CAM plants, they all go through the Calvin cycle. One other way to think about how these are separated is in terms of where the CO2 goes. In the C4 plants, the carbon dioxide intermediates are fixed and put someplace else. They're spatially pulled off and stored someplace else for use later on. Whereas in the CAM plants, they're temporally displaced. So the inorganic molecules are still there in the same place, but during the time of the day, they're just stored there until we actually get the energy from the light reactions. So CAM is temporal or time displacement, and C4 are spatial, where they're just stored in different parts of the cell. Let me shut this down.